Hello everybody, once again in another video about me talking about the process of protein synthesis. In this video, I'll be talking about amino acid activation, but I'll talk more about translation. Anyways, before I start the lecture, I want to share a small tip which have been useful to me in my exams and in my tests, quizzes, which is sometimes when I stress out, I struggle to remember what comes first, translation or transcription. Therefore, I use the alphabetical order. So if C comes before N, then transcription comes before translation. And that's how I remember the whole steps or stages in protein synthesis. Anyways, let's begin our lecture for today's presentation. The lecture outcomes are amino acid activation stage, the process of translation, the definition of the term polysome, triplet codes, codons, and anticodons, and of course, a quick summary for conclusion. Basically, amino acid activation is the second stage in protein synthesis where it occurs in the cytoplasm. In this stage, amino acids are activated by combining with short lengths of a different sort of RNA called tRNA. While tRNAs are relatively short, they are long enough to be folded into the shape of a leaf, specifically a clover leaf. Their most important feature is that there is a different tRNA for each of the 20 amino acids involved in protein synthesis. So each amino acid has its own type of transfer RNA. At one end of each tRNA is the site where the particular amino acid can be joined, and the other side is, in the other side, a loop, a folded loop is formed. Here, a sequence of 3 bases forms an anticodon, and this anticodon is complementary to the codons from the messenger RNA that codes for the specific amino acid. Pardon. The amino acid is attached to its transfer RNA by an enzyme, and these enzymes too are specific to the particular amino acids and types of the tRNA to be used in protein synthesis, of course. The enzymes is a further way of ensuring the code is implemented correctly and the amino acids are used in the correct sequence. So that's the role of the enzymes in amino acid activation. Now for stage 3, which is translation, this stage occurs in ribosomes, unlike stage 2, which is amino acid activation, which occurs in the cytoplasm. Stage 3 translation occurs in the ribosome. You can see that here that the ribosome consists of a large and a small subunit. Both parts of the ribosome are both parts of these ribosomes are involved in the process of translation, as you can see in this slide. So first of all, on arrival at the ribosome, the mRNA molecule binds to the small subunit at an attachment site. In this position, there are 6 bases, 2 codons of the mRNA exposed to the large subunit at any time. The first 3 exposed bases, which are codon of messenger RNA, are always AUG, a molecule of transfer RNA with the complementary anticodon UAC forms hydrogen bonds with this codon. The amino acid methionine is attached to this transfer RNA molecule. And then a second transfer RNA molecule bonds with the next 3 bases of the messenger RNA molecule, bringing another amino acid alongside the methionine molecule. Which amino acid this depends on the second codon, of course. Of course, while the 2 amino acids are held close together within the ribosome, a peptide bond is formed between them by condensation reaction, and this is catalyzed by an enzyme found in the large subunit, which I don't know if you can see, but this is the subunit here. Anyways, while the 2 amino acids are held close together within the ribosome, peptide bond is formed by the condensation reaction, as I said before, from the by the catalyzation of the enzyme found in the large subunit. And now the ribosome moves along the messenger RNA molecule in the direction of 5 to 3. and the next codon is read. At the same time, the first transfer RNA with the anticodon UAC leaves the ribosome minus its amino acid. Then a third transfer RNA molecule bonds with the next codon of the messenger RNA molecule, bringing another amino acid alongside, the second amino acid residue of the dipeptide. Immediately, a peptide bond is formed between them by condensation reaction, and a tripeptide has been formed and starts to emerge from a hole within the large subunit. Again, the ribosome moves along the messenger RNA molecule, and the next codon is read. At the same time, the second transfer RNA molecule leaves the ribosome minus its amino acid. Then, a fourth transfer RNA brings another amino acid to lie alongside the third amino acid residue, whilst three amino acids are held closely together. Another peptide bond is formed. By these steps, constantly repeated, a polypeptide is formed and emerges from the large subunit. Eventually, a stop codon is reached, and this takes the form of one of the three codons, UAA, UAG, or UGA. At this point, the completed polypeptide is released from the ribosome into the cytoplasm. And I just want to mention a quick thing or a quick note that gene mutation actually occurs because the stop codons, which are UAA, UAG, or UGA, are not actually present in the process of translation, which makes the process of translation continue further until it becomes unstable and it won't stop until it finds a stop codon. So if there is if the stop codon is not there, then it will immediately start to process more, therefore causing gene mutation. Anyways, let's talk about polysome. So many ribosomes occur freely in the cytoplasm, and these are the sites of course, protein syntheses that are to remain in the cell and fulfill particular roles there. It is common for several ribosomes to move along the mRNA at one time. The structure consists of mRNA, ribosomes, and the long protein chain. And this whole thing is called the polysome. Other ribosomes are bound to the membranes of the endoplasmic reticulum, known as the RER, or rough endoplasmic reticulum, and these are the sites of synthesis of proteins that are subsequently secreted from cells or packaged in lysosomes there. Now, triplet codes, codons and anticodons are so basically the sequence of bases along DNA molecules is called the genetic code, and this sequence ultimately determines the sequence of amino acids from which polypeptides are constructed. So the sequence of bases in the DNA molecule is transcribed to a complementary sequence of bases in the messenger RNA. DNA codes are referred to as triplet codes, and those in mRNA are called codons. So in DNA, they are known as triplet codes, and in messenger RNA or mRNA, they are known as codons. And then, free amino acids in the cytoplasm attached to specific transfer RNA molecule. There is one transfer RNA molecule for each of 20 different amino acids, as we have mentioned before, and the triplet of bases of the transfer RNA molecule is known as an anticodon. So for DNA, it's known as a triplet code. For RNA, it's codons, and for mRNA, it's codons, and for tRNA, it's anticodon. Anyways, in ribosomes, tRNA anticodons align with RNA codons or with the mRNA codons by complementary base pairing, and this brings amino acids side by side, and peptide bonds are formed between them. And eventually, a polypeptide molecule is formed. Now we'll go to the summary of the whole presentations and videos of everything. So basically, there are three stages to polypeptide and protein synthesis. First of all, in the nucleus, a copy of the genetic code of a gene is made in the form of a single strand of messenger RNA in the stage called transcription. The messenger RNA passes out into the cytoplasm, and in the cytoplasm, amino acid activation occurs, Each of the amino acids is attached to a specific tRNA molecule. Finally, a new polypeptide is assembled when the information of the messenger RNA is translated, read in a ribosome. The transfer RNA molecules with their attached amino acids are aligned in the order given in the code. Peptide bonds are formed between the amino acids later. So the three stages are transcription, amino acid activation, and translation. And of course, tRNA molecules occurs in the cytoplasm, and these molecules have a particular triplet of bases at one end, known as the anticodons, as we have mentioned before, and a point of attachment for specific amino acid at the other end. The role of tRNA is to pick up free amino acids and bring them to the messenger RNA and the ribosome. Complementary base pairing between the codons of mRNA and the anticodons of the tRNA results in the original DNA code being translated into an amino acid sequence in the process of polypeptide, or should I say protein synthesis. Anyways, thank you for listening, and I hope that I've helped you in your studies for your exams, and I wish you best of luck.